Hello, my name is Camryn. Today is August 18th, 2025, and this is my week 11 video summary. This video summary is talking about how DNA sequences will help shape proteins. But to begin this, we first need to know what exactly the non-coding DNA mystery is. So the non-coding DNA mystery is the mystery of while we figured out the entire sequence of our DNA, a very small percent, only about 3%, actually codes for the proteins. And we didn't know what the rest of it did, so we called it non-coding DNA. Sometimes we called it junk DNA or selfish DNA. But it all meant the same thing because while we had tests to show that it's folding certain ways and things like that, we didn't know that the DNA sequences were actually what was causing the folding. So this was until the genomic code where we learned that the junk DNA actually contains the sequences that control how it folds. And then we go into this article which gives us evidence of that fact of the sequence controlling the folding. One of the first forms of evidence we see is that coding sequences and the bases are very unevenly distributed throughout the human genome. So, like I said, protein coding is only about 3% of the genome, and we see two different forms of that genome. We see the high GC or GC rich, or the poor GC or GC low areas. Um, GC obviously stands for cytosine and guanine, and those high GC areas and those rich GC areas are going to be where there's a lot of guanine, a lot of cytosine, a lot of those base pairs together. We see that along the H3 isotures, and we call these areas with this high level the genome core because it is that open chromatin that is located in the center of the nucleus. On the other hand, that low GC, GC poor areas, you're going to see more adenine and thymine within it to be forming those genome poor areas. We also call it the genome desert. And that's going to be that closed chromatin, and we're going to see it on the peripherals of the nucleus, so it's not in that core area. We also use modern technology like the Hi-C test to kind of confirm this with two compartments. We call them compartment A and compartment B. Compartment A shows that active gene, which is that rich open chromatin, which is our GC high areas, with the B compartment being the inactive gene and that poor genome with GC low areas that's on that peripheral with closed chromatin. We also discover topologically associated domains, or TADs, along with which are folding areas that are stable folding units within the sequence, and the lamia associated domains, or LADs, are confirmed, and those are also GC pore regions. Another thing that's evidence for this are isotures, which are long regions of the DNA sequence where the GC is uniformly split. So it could be a long sequence of very rich GC DNA, where it'd be a bunch of GC bases, or it could be a GC pore area, but it's all kind of uniformly spread out in this area. An example of this is chromosome 21. But it helps us view the shapes of a GC poor area versus a GC rich area. So using the dot plot, we could see the GC profiling more. And did we see in GC poor isotures where it's more A and T's, we see very flat, like the LADs, which is what we're seeing. And then the TADs, which are GC rich, we see a bunch of peaks and valleys to show variation in folding. We also have evidence of trinucleotide repeats being unevenly split, so the AAA, triple Ts are obviously in GC poor areas because more adenine and thymine, while the triple Gs and triple Cs are more seen in genome or GC rich areas because, of course, it's guanine and cytosine. The formations of LNDs and LADs and TADs are also just evidence to support this because the LADs have that B compartment GC poor because they're all flat, while the TADs are that GC rich with the valleys and dips that show stable bending through a two-step process of molding a cohesion to make those A compartments. Overall, I think this essay and article is really important to show not only how much we've grown in knowing our DNA and our structure, but how much further we can go with it. Because 20 years ago, we didn't know anything about our structure beyond we have the genome and we had no meaning behind it. But this is a beautiful step of how much we are learning about our genome and also how much further we have to learn about ourselves and who we are.